Hello. In this presentation, I will tell you about the generative modeling approach for in silico experiments. The original problem was which surface topographies to use in the next experiment based on the collected screening data. And this problem can actually be considered as part of the concept of autonomous system for biomaterials discovery. When we have accumulated experimental data, experimental screening data, we want to analyze the data and design new materials for the next physical experiment. And the question is how to apply machine learning to perform these two steps. According to the literature, there are basically two approaches to address this problem. The first is modeling self-surface topography interaction. Here, the analysis of the data is made by modeling the relationship between the topography properties and the cell properties. And here are some examples from the literature listed here. And the topography design here is made by adjusting specific topography properties according to the influence based on the model that we made. So to optimize the cell response, we adjust specific topography properties. The second approach is the evolutionary approach encountered in one paper. Here, the analysis does not include any model of the relationship between the topography and cell. Rather, we select best hits according to some cell response that we want, and we perform crossbreeding of the best hit topographies in the hope that it will lead us to the optimized cell response in the next physical experiment. Well, taking a closer look at the first perspective, the modeling self-surface topography interaction approach, the ultimate goal here is to discover the relationship between two high-dimensional spaces based on screening data, based on training pairs, where the input parameter space or the topography design space is somehow related to the outcome space, being the cell images space. And the question is, how can we model this relationship? When we talk about space of images, we assume that an image X can be considered as a point in the space X. For example, if we have an image 128 by 128 pixels in one color channel, being the grayscale, this object can be considered as this object is actually a 16,000 dimensional vector and is point in the 16,000 dimensional space. And taking an example from the Muniz dataset, the point x1 being the 16,000 dimensional vector can be considered, can be visualized as an image for our purpose, but actually it's just a vector. But in this example, the actual dataset size is 28 by 28 pixels, but this can be any image and any dataset of images of any size. So when we talk about modeling self-surface topography interaction, how it is done currently based on the literature, usually we do regression or classification of key cell features instead of the original images. So we model or we predict certain cell features of interest, such as nucleus form factor, cell orientation, or biomarker expression, based on input topography parameters, which are, for example, number of primitives, shape of a primitive, surface density, and others. And to optimize the cell response in the next iteration of experiments, we adjust specific properties of the topographies, PJ. So what is special about these models? All these models are point estimate models, meaning that we predict an average cell response Fi. For example, based on a single topography, we predict an average nucleus form factor that we can expect based on our data. And secondly, machine learning is used as a tool to analyze data and find correlations, or we can say like a calculator. And the question is, can we leverage machine learning more effectively, or can we take some more advantages that machine learning has to offer? And the question is, for example, can we use machine learning to simulate the experiment? That means that given a topography, we would like to generate possible cell images that can be generated based on that topography in real life. And vice versa, can we use machine learning as a tool for biomaterials discovery? And here we give a cell image and we would like to generate topographies that could have led to this image. So given a cell image with some desired properties, we would like to understand which topographies could have led to these desired properties. And taking these topographies, we pass them to the next physical experiment, and we expect to see these desired properties more often than in the previous physical experiment iteration. Here we can ask ourselves, why would we want to simulate the experiment in the first place? And there could be several reasons for that. Firstly is we might want to observe a full outcome, being the cell image, in response to topography, because it was easier for an analyst to judge whether the outcome is desired or undesired. We can see multiple properties according to one model. So instead of having multiple independent models for each of the feature of the cell, we may want to observe the full outcome and to see these features in the same picture. The second reason is such a model could potentially speed up biomaterials discovery, since only a fraction of the parameter space or the topography design space can be covered by physical experiments in reasonable time. While having a simulation model, we can run the experiment in silico many times, and as a result we can select the topographies for the next iteration of physical experiments more thoroughly. And the third reason is that we may want to account for uncertainty in experiment. So instead of outputting or predicting some average responses, average nucleus round factor, for example, we may want to output probability distribution instead of point estimates. In other words, we may want to account for variance in the output. In this, here we assume that topography actually leads to a distribution of cell images. And this is, by the way, supported by the fact that given the same topo chip, the same topography is placed multiple times, and it leads to maybe similar but different pictures of different cells. And we want to account for that in our model. And vice versa, we assume that a cell image could originate from a distribution of topographies, which again can be similar, but they could be different in some way. And the proposed approach to address this issue is to build a deep generative model. And the key steps here are, firstly, representing high-dimensional spaces X and P in low-dimensional latent spaces. And the second step is modeling the cell-surface topography relationship in these latent spaces instead of modeling them between the original spaces X and P. As a result, the model will produce three types of probability distributions. First is probability distribution of images based on the topography, on a given topography. Secondly, probability of topographies given a cell image. And thirdly, and also a convenient way to make curious, is the probability of a distribution of topographies given some feature of a cell image. So, for example, given a nucleus factor, form factor, we would like to generate different topographies. So, in more detail, the application scenario of such model could be twofold. Firstly, the in silico experiment. That means that we would like to sample from the distribution of images given a topography. And the query here is generate different cell images, X, in response to a given topography P. The second application scenario is the in silico topography design. Here we would like to sample from either probability of P given X or of P given Fi. And the queries are as follows. Generate different topographies P that could have led to a given image X. And secondly, generate different topographies P that could result in a given value of Fi, for example, nucleus form factor. Taking a closer look at these steps. The the first step is to represent the high-dimensional spaces X and P in low-dimensional latent spaces. Here we assume that there is a low-dimensional representation Z of a cell image X. So for example, if we take pictures of 128 by 128 pixels, it is a 16,000 dimensional vector, but in fact we may assume that the cell image itself can be explained by a much fewer number of factors, such as shape, nucleus size, orientation, and a couple of others. But we can assume that a maximum of 10 dimensions is needed to fully describe the variation in images. So we take a latent space, a low-dimensional latent space, for example, 10-dimensional, and our goal here is to train a decoder, which is a mapping from Zx to X, to be able to sample from X, so to generate pictures by just sampling from a lower dimensional distribution. So taking a 10-dimensional vector, we generate images from X. And accordingly, we do the same for parameter space, for topography design space. We would like to train a decoder TP to be able to sample from P to generate topographies by just sampling from LP, from low-dimensional latent space. But when we talk about latent space of images, it should be noted that it is usually a bidirectional mapping. So we have both a decoder and the encoder. The decoder aims to map a point from the latent space Zx to some distribution or a region in the space X. So given a vector of like a 10-dimensional vector, we map it to some distribution of images. The distribution itself of P of X given Z can be very narrow. So this usually contains images that are really close to each other, they're really similar to each other, but still it is not a point, it's just, it is a distribution, and this is the essence of the generative modeling. Secondly, we have an encoder that does the inverse mapping. It takes an image, a point in the space X, and maps it to the distribution or some region in the space Zx. It can also be very narrow, it can almost be a single point, but still it is a distribution by design. So we have some mean and some variance, at least if we talk about two parameters, a distribution with two parameters such as normal one. An example of these mappings with the NIST dataset. Suppose that we have a two-dimensional latent space Zx, then consider what a decoder does. We take some point in this latent space Zx, which is in our case a two-dimensional vector. It has only two values, two numbers, and we pass it to the decoder, and it outputs some distribution of our images. Or for example, if our image is a 16,000 dimensional vector, then it outputs a distribution, a 16,000 dimensional distribution with some mean and some variance. And the image, if we take samples from this distribution, from this small region, we will get images that are slightly different, but in general are close to each other. It's not guaranteed, but it's usually like this. So it is the same, basically the same picture, but with some variation. And if we take another point in the space, in the latent space Z X, it generates another small region in the space X, and by taking points in the space X, we just observe maybe similar, maybe not very similar, but images from this space. The encoder is the inverse mapping, and here we just take a point in the space X, being the images, so for example X1, and we pass it to the encoder, and as a result we get some distribution or region in the space Zx, which is a distribution over a two-dimensional vector. It could be a distribution like this, so some mean value, two-dimensional, and a covariance matrix. So suppose once we have this latent representation learned, one for the space of images and one for the space of topographies. The key idea here is to model the relationship between the data sets. not between the original spaces, but between the latent spaces. So because we already have the decoders that are able to generate objects from each of the dataset by just sampling from this small distribution. And the only thing that needs to be done is modeling the relationship between these latent spaces. So how can we model this relationship? The first step is we assume that topographies P may influence cell images through visually discernible and measurable cell properties Fi. For example, nucleus form factor or cell orientation. And in turn, a cell image X is a combination of the cell properties of these cell properties and some residual variation. So for example, if we assume that a cell image can be represented by only two factors, nucleus form factor and cell orientation. So if we're interested in these factors, and then we assume that all the rest variation, like shape, maybe some elongation, it goes to the epsilon, to the noise. And we model the influence of topography exactly on these selected factors of interest. So the second step, once we have assumed that the image of a cell is explained by a couple of factors of interest, or features, we would like to factorize the latent space X by cell properties of interest and noise. So we represent the latent space as a dot product of subspaces, where ZFI subspace corresponds to the property of Fi of a cell image, and the last subspace, the Z epsilon subspace, captures the residual variation in X that is not explained by the feature Fi. And to generate a cell image, we now don't take only one point from the space X. We take a point from the space ZF1. We take a point simultaneously from the space ZF2, ZF3, and Z epsilon. We take these 4 points, we pass these 4 points to the decoder, and as a result, we get some distribution or a small region in the space X. Yes, an example of the factorized latent space can be shown again with the MNIST dataset. Let us assume that an image of a digit can be represented by just 3, the handwritten digit can be represented by 3 features. The digit itself, whether it's a 0 or 1, etc., which boldness it has, and a curvature, so how curved it is. Then we assume that these factors are independent of each other. So we can represent the latent space as a dot product of independent factors of variation. So here we may take a point from the space F1, for example, it's a 9, and by traversing this latent space, we can generate different images of different digits. By traversing the latent space corresponding to the boldness feature, we can generate, for example, the same digit but different boldness, and the same for curvature. So now, once we have this latent space split into different factors, the different subspaces corresponding to these factors, we would like to reuse the latent spaces, subspaces ZFI, as part of the latent space for topographies. And this is the second key idea here. So now LP is also represented as a dot product of subspaces ZF1, ZF2, ZFN, and the residual subspace L epsilon. The only thing that changes is the interpretation of these latent subspaces. Now the subspace ZF1 represents the influence of topography P on the cell property F1. So ZF1 and F1. And the subspace L epsilon now captures the variation in topographies that is not related to their influence on X. For example, if there is some feature of topography, like, let's say, a density, and we know, or it follows from the data, that density of the topography of these pillars does not influence cells anyhow, and we know this for sure. Then all the variation of the density will be captured in this space. So because this has no relation to the with the cells. So now let us consider how such a model can be applied the application scenario in this case. And the first application scenario is the in silico experiment, where we would like to consider the distribution P of images of X given P topographies. Here we take some topography, and the query would like to generate the images Xi, which can originate from this topography, which can be generated if we ran the experiment with such a topography on the chip. So what happens? We pass this point P from the space P to the encoder P, and as a result we get some regions in the latent space LP. So we get some region in the space L epsilon, but we are not very much interested in it. We get some region, blue region in the space ZF1, and this region corresponds to a distribution of ZF1 given P. We get some region in the space ZF2, and we get some region or a distribution in the space ZF3. Now we would like to generate possible cell images, and to do that we take some point in this region. We take some point in the region ZF2, and we take some point in this region ZF3, and these regions correspond to a single topography. And also we would like to take a point from the space Z epsilon, which corresponds to the residual variation in images, some noise variation. We also take one point, and then we pass this we pass these 4 points in the decoder X, and as a result we get some region in the space X. And by taking a single value, for example a single value from this region, we get a cell image that is an output of the model, and this cell image is basically the output of our in silico experiment. But we would like to generate different cell images to see which different cell images can be generated based on a single topography. To do that, we take another point from this region, another point from this region, another point from this region, and also another point from the distribution of Z epsilon. And we pass these 4 points to the decoder again, and we generate another small region, or maybe not small, some distribution in the space X. Here we also can take a single point, and we get another picture, another image of a cell. And so why do we take a single point from each of the region? Because again, usually the points in this space, in this distribution of P given Z, they are quite similar. So to see different images of cells which look different, we may want to sample multiple times from ZF and take only at least 1 point from each of these small distributions. And as a result, we get a collection of Xi, which is the output of our in silico experiment. The second application is the in silico topography design, which is the inverse mapping, inverse traversal of the model. Here, the query is to generate different topographies that could have led to a given cell image with some probably desired properties. So we take a single image, we pass it to the encoder X, and as a result, we get the distribution within the subspaces ZF1, ZF2, ZF3, and Z epsilon. So we get some subspace, or so we get some distribution or a region in space Z epsilon, but again, we are not very much interested in that. We get some region in the space ZF1, ZF2, and ZF3. And the region, for example, here, the orange one, corresponds to a distribution of ZF1 given X. Then we would like to generate topographies. We take a single point from this region, and from this region. And also, we take a point from the space L epsilon, which corresponds to the variation in topographies that is not related to its influence on images, on cells. So taking these 4 blue points, we pass them to the decoder P, and as a result, we get some distribution in the space P, and taking a single or multiple points from this distribution, we get topographies. This could be, for example, images as well, but this could be some parameter representation. So we get topographies that, according to our model, could have led to such an image. So to get different topographies that could have led to this given image, we sample from Z space multiple times, from ZF1, ZF2, ZF3, and L epsilon, from LP space, I meant. So this is depicted as well here. So we take another blue point here, another blue point here, another blue point here, and another blue point here. We pass these 4 points to the decoder P and get an outputted distribution over topographies. So again, we sample from Z to get different small distributions in the space P to get visually different, or not completely different topographies, to maximize the differentiation in topographies that, according to our model, could have led to such an image. And the third application is in silico topography design, is given the desired properties of cell images. Here we only consider the sum of the properties that we are interested in. For example, if F1 property is the nucleus form factor, and we are interested, let's say, in some high values. For example, 10 is a high value of a nucleus form factor. We pass the value 10 to the model. The model is able to generate a distribution of ZF1 given F1 equals 10. So this can correspond to such an region in the space ZF1, and we only now would like to sample from this region to generate different topographies. But other subspaces, in this case, we're not interested in them. We just sample from the prior distribution, from the distribution ZF2, distribution ZF3, from the full distribution. So we take some point here, some point here, some point here, and some point again from the L epsilon distribution. And as a result, again, we get some small distribution of topographies corresponding to this value of feature Fi in the space P. And by sampling many times different points from ZF1, ZF2, ZF3, and L epsilon, we get different small distribution, and as a result, we can make a collection of topographies that could result, according to our model, in a value Fi. So to validate this idea, whether it can be applicable on some data, on some training data, I performed an experiment on a toy data set. So first of all, I created a toy data set. So I generated 50,000 images, artificial images of cells, which is a data set of X, and 50,000 of artificial images of topographies, or something that can represent topographies. Again, topographies may be represented by a vector or by an image, and it does not really matter. Here I consider topographies to be images just for convenience, to map images to images. So considering X, here the cells, the artificial cells, are centered around nucleus and they have 4 properties: roundness, elongation, nucleus size, and rotation angle. But rotation angle here is a feature that we are not interested in. We assume it to be the noisy feature. For the topographies, it is a single topography is represented by 9 shapes with fixed center locations and only 2 features of variation. These are roundness and radius. So considering again these features, if you look at the data set for X, roundness feature is how smooth the corners of the cell are. Elongation is quite an apparent feature, and the nucleus size, again, it's the size of this little orange circle in the middle. So this is the smallest nucleus size, and this is the largest nucleus size in this data set. And the rotation angle is the rotation in the inverse direction of the clock. So, but this is the noisy feature, and we are not interested in how the cell is rotated. We are only interested in these 3 features. The data set for P looks like this. So roundness, again, is the lowest value looks like a star, and the highest value looks like a circle. And so the radius is also a very apparent feature. This is the smallest value of radius, and this is the largest value of radius. So to build the model, I assumed that there is some relationship between P and X, and we needed to create artificial training pairs for the model. So I assumed that there is an influence of radius of the topography on the elongation of the cell. So the training pairs examples are presented here as well. So I passed to the model the pairs like small radius topography and a small elongation, low elongation cell, a low radius, a high radius topography and high elongation cell. And this is the only source of data that the model used. No, apart from the values of the features, which are also were consumed by the model. So the first step was to factorize the space Zx to represent images of cells. And here we can see that the space, the model was able to learn the disentangled, the factorized representation, later representation of a cell image, where ZF1 space corresponds only to the roundness feature, the ZF2 space corresponds only to the elongation feature, and ZF3 space corresponds only to the nucleus size feature. And the Z epsilon space corresponds to the residual variation, which in our case was only the rotation angle and some background noise. So so the model was actually able to capture that the last space should learn only the rotation angle. So if we color this space, for example, by a sign of elongation, rotation angle, we can see the clear structure here. But this is more about technical details. So as a result of this step, we're able to generate realistic, rather realistic images of cells by taking points from each of the spaces. 1 point here, 1 point here, 1 point here, and 1 point here. We pass these 4 points to the decoder and get some distribution of images where this is a single sample from this distribution. The second step was to reuse the latent subspaces, ZFI, as part of the latent space for topographies. And here, this is the most important concept of the model, where the latent subspaces are reused, but they change the interpretation. So here, if we take a closer look, for example, at the subspace ZF1, we can see that the model, if we encode some topography to the region in the space ZF1, which is probability of ZF1 given P, and this is the green distribution, we can see that actually any topography of any radius or any roundness will map to the full distribution ZF1, because ZF1 was initially structured according to the roundness feature of the cell. And since radius and roundness features of topographies have no influence on roundness of the cell, then there should not be any structure according to any feature of the topography. So each topography of different radius, of different roundness, will map to the full distribution ZF1. There is no distinction between radius and roundness, since they have no influence on roundness of a cell. In contrast, if we look at the second subspace, ZF2, we know that there is a connection between there is a relationship between the radius of the topography and elongation of the cell. And since the subspace F1 in the space Zx was devoted to the elongation feature of the cell, we expect that high radius topographies will map to the region of the space ZF2, which corresponds to high elongation region of the space of images. So here, for example, since the topography has some quite a high radius, it maps with the encoder of P. It maps to the region of the space ZF2, which corresponds to high elongation of the cell, as we can see here. And accordingly, if we take some, for example, low radius cell, low radius topography, it will map to some region in the space ZF2, which correspond to low elongation of the cell. And if we look at the third ZF3 subspace, there is also no relation, no structure according to radius or roundness, since there is no connection with the nucleus size. What is also interesting in this part is that the L epsilon part is L epsilon subspace corresponds to the residual variation in the topographies that has no relation to the influence on cell images. So since the roundness of the topography had no influence on cells on any feature, so f1, f2, f3. So there was no relationship tied to roundness. So the L epsilon space is structured according to the roundness feature. And now, once we have these components of the model trained, we can test how it could perform the queries that we wanted to perform. The first is in silico experiment, where we would like to sample from the distribution of images given a topography. The query here is to generate different cell images in response to a given topography. And by passing, for example, a high radius topography to the model, we expect to see high elongation cells, but which vary in all other features except for elongation, because high radius leads to high elongation, but there is no connection between radius and roundness or nucleus size. So what we expect to see, what we would like to see, is different nucleus sizes in the output, as well as different roundness of the output, which is also reflected here somehow. And also, rotation angle is completely different. So there is no model of the rotation angle, it's just in noise. And accordingly, if we pass to the model some low radius topography, what we expect and what we actually see is different cells, which all have low elongation, but they have different factors, different other factors, such as nucleus size. This is a small nucleus size, this is a large nucleus size, and different rotation angle and different roundness. But roundness is not very much seen here because the elongation is quite low. The inverse application scenario is the in silico topography design, where we want to generate different topographies P that could have led to a given cell image X. For example, if we're interested in high elongation cells, if this is a desired property of a cell, we pass this high elongation cell image to the model, and the query is to generate different topographies which could have led to this image. And as we can see, the model is able to generate different topographies which firstly look similar to the pictures from the data set. Secondly, they all have high radius, since this is the relationship, and also they are different in other factors. But in this case, the other factors is only the roundness of the topography, so we see this variation in roundness, but with the high radius still. So, talking about limitations and the future work of the current for the current framework, one of the assumptions that makes this approach limited is the independency of features Fi assumed. So I assume that an image of a cell is represented by number of independent factors such as elongation, nucleus size, and roundness. But in reality, these factors can be somehow related. Even in my case, if I take low elongation, the roundness feature can be less apparent than for high elongation. But still in my example, they were quite independent. But in real life, this could be a problem potentially. This is a weak point of the model, potentially. The second point is that the model is able to generate individual images of individual cells, but maybe we are interested in generating multiple cells, and this is the key. This is the most important property of a model. But such a model is not able to generate multiple cells because it's a much harder task for a generative framework. And here there is also a question: should we generate the cells iteratively, different pictures of different cells, and then some algorithm should put them together in one picture? But maybe it is superfluous, and this is excessive. I mean, redundant. We don't need this. And the third point is that P and X are considered as independent spaces. And the question is it possible, and whether we need it actually, is it possible to generate cell or cells on a topography? So as input, we take a picture of topography, and as output, we see the picture of the same topography, but with the cell placed on this topography. And again, the question is whether we need this or not. This is all for that presentation. Thank you for your attention, and these are the references for the presentation. Thank you.